Hello, I'm Jan Zrimets, and I'll present the paper titled Structural Representations of DNA Regulatory Substrates Can Enhance Sequence-Based Algorithms by Associating Functional Sequence Variants. So this conference paper presents the general framework of the DNA structural representations and discusses how it could be possibly used to improve different bioinformatic algorithms, but here I will mainly focus on the actual problem that they were developed to solve. This problem is related to horizontal gene transfer and prediction of plasmid mobility, which we have already studied in a previous published paper, and I will briefly recap here. The way bacteria pass a genetic material, such as for instance antimicrobial resistance genes, is by horizontal gene transfer. And the majority of horizontal gene transfer events occurs via the process of plasmid conjugation. So the main components of the conjugative transfer system on plasmids include the origin of transfer regions, which here I will just call transfer regions, which are located on the plasmid itself, as well as enzymes and proteins that trigger and facilitate the DNA transfer. Because the transfer regions are the places where plasmid mobilization initiates, they can be viewed as some of the most important elements for transfer, and they are the substrates for protein and enzyme binding, which form a complex at this region that unwinds the DNA and starts the DNA transfer. So, based on the amino acid conservation of the main enzyme, called relaxase, different plasmid mobility groups were defined, and they were also shown to define plasmid host ranges, meaning that it's important to be able to predict these mobility or mob groups. On the right figure, 4 mob groups are shown, F, P, Q, and V, as well as the different transfer regions across different plasmids. And they are all aligned to the main enzymatic nicking or nick site where transfer initiates. So these regions are approximately 220 base pairs long, carry multiple binding sites, hairpins, and inverted repeats. But we also see they have approximately conserved properties across the mob groups. If we look at the general outline of these regulatory transfer regions, they are very similar to the regulatory regions such as promoters. They both carry a main enzymatic site, marked red, as well as multiple upstream binding sites for accessory proteins. In both types of regions, especially at the enzymatic site, a DNA melting bubble must form to give the enzyme access to the DNA. And this melting bubble formation is affected by multiple things such as neighboring sequence, nearby protein binding, and thermodynamic stability effects. And also, we found that the different binding sites in the transfer regions carry multiple conserved DNA structural properties that facilitate recognition and binding of the protein to the DNA. And these include different physicochemical and conformational properties, such as DNA stability, flexibility, the amount of helical repeats, which all contribute to protein binding. So the general DNA structural properties important for protein DNA binding are further reviewed in a supplementary table accompanying the conference paper. So trying to discriminate the mob groups based either on their sequence or structural properties, we see that based on structural properties we can achieve significant discrimination of mob groups, which is actually not achievable just by looking at the nucleotide sequence. And if we build machine learning classification models, we are able to very accurately predict different mob groups. So if we look at which parts in the transfer regions are actually important for discriminating the mob groups, we see that with the structural properties, different locations across the whole transfer regions are conserved, whereas with nucleotide sequence, we only have a conservation of properties around this main nicking site. So this shows that with the structural properties, we're able to use a much larger amount of information stored across the whole region to determine the mob groups. Okay, so considering that not many specific tools exist for finding and mob typing transfer regions, and that they have these conserved structural properties that could be potentially used to search for them, we then try to develop an algorithm to find and type these regions. For searching regions in large sequences, alignment algorithms are usually used, and these require a distance measure that can be used to find the most similar regions in the target sequence to the query sequence. To develop a structural distance, we needed a way to encode DNA, so a structural representation of DNA, which could be used to compute the distance between two sequences. And this development is presented in the conference paper. First, we fused 64 of the most frequently used DNA structural property models into compact DNA representations. Since between 1 and 4 neighboring nucleotides around the central nucleotide have the most effect on its structural properties, we encoded k-mers of sizes 3, 5, 7, or 9 base pairs. So for all permutations of these k-mers, we computed the structural properties and performed dimensionality reduction with PCA, where up to 18 principal components were used to retrieve most of the data variance. We further compacted this data with clustering to a different amount of clusters ranging from 4 to 256 clusters. The figure on the right shows the first two principal components with k-mer size 5 and 16 cluster centroids. So this way, k-mers were structurally encoded into what we called S-mers. So scanning a DNA sequence and converting its k-mers into S-mers gives us a structural representation of the sequence. Importantly, when we recovered the key distinguishing properties of the first 6 structural components, we found that they indeed reflected the main properties involved in protein-DNA interactions, such as thermodynamic stability, conformational stability, and flexibility. We next explored if the new representations could encode groups of conserved functional DNA motifs, and could encapsulate DNA motifs more compactly than bare k-mers. For this, we used transcription factor binding site motifs and found that the compression ratio of functional DNA sequence motifs could be as high as 3:1. The most promising results were obtained with a number of clusters corresponding to 6 to 8 bits information, so 64 to 256 clusters. And these representations could compress functional sequence variance by around 30 to 50%. We then explored how many sequence variants of a motif, or whole regulatory region, each instance of the structural representation encapsulated. And we found that representations of transcription factor binding site motifs with S-mer size of 7 base pairs encoded on average 2 to 10 sequence variants. But with whole transfer regions, this increased 32-fold, so a single instance of a representation could encode from 10 up to 2,000 sequence variants of a region. Then finally, a structural distance measure could be developed, which was termed the S-distance. This was defined as the sum of Euclidean distances between the cluster centroids of consecutive S-mers in two different structural representations. We then analyzed and compared the performance of the S-distance to the nucleotide sequence p-distance, which is the Hamming distance scaled by the sequence length, when trying to discriminate positive or negative examples or among different mob groups. And this was done across different sized parts of the transfer regions, so from the central 40 base pair enzymatic substrate to the whole 220 base pair regions spanning multiple different binding sites. We found that with the shorter 40 base pair parts, we got significant discrimination of both positive negatives as well as mob groups, whereas for instance with the whole regions, only the structural representations were able to successfully, so significantly, discriminate among the different functional groups. So the structural representations seem to increase the resolution between functional groups of sequences, which enables them to resolve regulatory DNA more accurately than bare nucleotide sequence. Okay, so now we had all the elements required to develop the structural alignment algorithm for finding and typing transfer regions. This included pre-computed encodings to convert a DNA sequence into a sequence of S-mer cluster centroids, as well as pre-computed distant matrices between the S-mers, both of which enabled faster execution of the algorithm. But besides this, we also developed a function to estimate the statistical significance of the S-distance scores. This was done by estimating the permutational p-values at different significance cutoffs and then curve fitting on this data to obtain a continuous function. As a note, this is the same algorithm as in the paper, just the definition is slightly expanded to include formally defining that structural representations are computed for both query and target sequences. When comparing this algorithm to using the p-distance as well as to BLAST, we found that it achieved improved performance both for locating transfer regions within one base pair of the nicking site, as well as for typing mobility groups and subgroups. The main improvements seem to stem from an increase in recall at a relatively constant precision, so the algorithm is able to recover a larger amount of the correct transfer regions compared to the sequence-based approaches. So the results I'm presenting here are actually from a related paper currently available on bioRxiv, where the new algorithm is used to type the transfer regions across thousands of plasmids. We recovered a large amount of the transfer regions which are expected according to relaxase enzyme typing, but also we find that twofold more plasmids actually carry these regions, which points to a twofold increase in putative plasmid mobility. and the algorithm is quite accurate at retrieving the mob groups according to the relaxase enzyme typing. But most importantly, we find that over half of the mobile plasmids carry multiple transfer regions from multiple mobility groups, which shows that they carry different transfer mechanisms that could potentially enable them to transfer among different host ranges. A very important finding is also that the amount of transfer regions per plasmid seems to correlate with the amount of antimicrobial resistant genes found on the plasmid. So these multiple transfer regions might present a mechanism for the observed quick spread of resistance from the environment to humans. Okay, so to conclude, I showed that the structural alignment algorithm can be used to find and mob-type transfer regions. And this is a nice showcase for the structural representations and S-distance, which could potentially be used to enhance other bioinformatic algorithms, such as, for instance, phylogenetic analysis, motif identification, or de novo design of DNA substrates. For instance, one combined sequence and structure approach that is shown in the figure, and which is based on evidence of how proteins actually recognize and bind their DNA sites, is to use structural representations to locate the broad portion of regulatory regions and combine this with finding finer, more specific parts, such as motifs or inverted repeats, down to the very small, exactly conserved sequences, in order to increase the accuracy of locating or typing specific regulatory regions. So with that, I acknowledge all the people and institutions that have helped me with this research, and also thank you for your attention.